In the first section, we talk about the history of cell biology, then we talk about the development of the cell theory. Now we're going to talk about how this microscopy allow us to identify all these different cells. And we're going to talk about the different sizes and variations of the cells and how we have to develop better microscope to study even in more detail those cellular structures and the different type of microscope that are used. Okay, so let's start. Now let's talk about cell variation. So the cells vary in sizes and shapes. And here in your textbook, there is a lot of examples. So if you took anatomy and physiology, you might know that we have some neurons that can be very long. Yeah, so the axons are very long. They can be a few centimeters or almost close to a meter in some of our legs to send the electrical signals as fast as possible. But most of our cells are really tiny, and we have cells in unicellular organisms or in our blood that have different shape, but they are regular. Generally, uh, they have spherical shape. However, this is not the only case. Yeah, we have many cells that can have multiple shapes, and I'm going to show you a video in the following slide that talk a little bit of that variation. If you are interested on weird animals, yeah, in weird cells, protists are an example of an incredible diversity. Yeah, they have some of them that have develop, yeah, that they have a bulb structure that look like ice, yeah, with a single cell. How is incredible, yeah. So I'm going to cover some of those, and I'm going to show you more videos in class about this variation. Again, key idea here is that cells, they can display a huge variation, both single cell organisms as well as in multicellular organisms. Multicellular organisms have a lot of variation also for different functions. It's very different a neuron to an epithelial cell. So now I wonder you think about this question that we will solve in class is why don't we find cells of the size of an elephant? Yeah, it's an amazing question, very simple for some of you, that it could appear some variation of that in the test. And I wonder you think about it. I'm going to show you cells that we could reach a few centimeters, but why we don't have cells of the size of an elephant, or do we have cells like that and we just don't know about it? Okay, so think about it, and we will talk about that in class. Okay, maybe you already thought about the question, and I'm going to give you a clue of how to answer that question. And I'm going to show you a video of giant cells. And there are many of them. I'm just going to show you an example of an organism, of a type of algae that had an incredible giant cell. Okay, and later during the semester, we're going to cover and explain some of the things that this adaptation that have this organism that is actually a giant cell.
At first glance, you would probably have no idea that Caulerpa algae are individual cells. Individuals in some species can grow to carpet a square meter or more, and they sure don't look like your average cells. Each has a complex structure that consists of a stem, fronds that look like leaves, and root-like parts that anchor it in place. The reason a Caulerpa can grow so large is because it has loads of nuclei to keep all parts of it functioning properly. Caulerpa species also use a process called cytoplasmic streaming, which is essentially a plant's solution to a circulatory system. They always keep their cytoplasm moving in an organized fashion, which allows them to quickly transfer molecules and other structures around the cell. Lots of scientists have studied these algae over the decades. Researchers were particularly interested in learning more about how they get their intricate structures, because it was long assumed that that kind of complexity required multiple cells. Now, they believe it all boils down to when and where different genes are active. For instance, research published in 2015 showed that different genes are active in the nuclei in different parts of the cell, and these seem to be telling those regions what shapes to be. This has deepened our understanding of how all cells figure out their final shape and structure, and it's actually calling into question whether any plant is truly multicellular. You see, some researchers argue that all plants should be thought of as single-celled organisms because their cells are interconnected and communicate with each other through channels called plasmodesmata. And if cells are super connected, Are they really separate cells? It's an intriguing question anyway. While humongous cells like Caulerpa are the exception, not the norm, because of their size, they've given researchers throughout history the incredible opportunity to dig into the typically microscopic world of cells. Because of that, they can help us understand the inner workings of all cells, big and small. In fact, you may have noticed that all these giant cells have something in common. They're all found in water. While it is possible for a large cell to exist on land, they're usually living in humid areas that have recently received heavy rains. Life in the water means less stress for cells of any size. No drying out, nutrients and waste products easily diffuse in and out, and the fluid pressure gives some extra support to their wall.
Okay, so far I have shown you that cells have different sizes. Here you see an schematic of two big cells, yeah, and here you see that are eukaryotics, and this one that is very tiny, that is prokaryotes. And if you remember, if you see here, this 10 micron, this symbol, yeah, we're going to cover that if you don't remember them. Yeah, so the idea that these cells, they have different shapes, they have different sizes, and even the structures, they are very tiny. So to study that, there was a field that create this revolution that is called cytology and study all these cellular structures and these different optical techniques. And in the following slides, we will talk about one of those optical techniques, that is microscopy, that will allow us to study all these structures that have a very small size. To start that, I want that you think about all the different biological structures, and we're going to organize based on size. Okay, so let me show you an example in the following slide. Okay, now I want that you arrange these biological structures based on their size, from the smallest to the largest. So here, what do you think the blood vessels, the nucleotides, these red blood cells? I want that you guess which one are the smallest one and which one are the largest. And if you want, if you feel confident, you can even guess the sizes, yeah, more or less. What do you think that is the size? It could be centimeters, it could be meters, it could be microns, it could be Armstrongs, it could be millimeters. I, this doesn't have to be perfect. I just want that you start thinking about all these different sizes that have different biological structures. One clue, you can organize it based on atoms, then go to molecules, organelles, then cells and tissues. This can help you to organize it from the smaller ones to the largest one. Okay, so organize it, and in the following slide, I will show you the answer. So let me show you the answers. Here you can see all of them. You can go from atoms, like here, to big tissues that are close to a centimeter, and cells and organelles that they could that are here that are in different measurements. So we can go from nanometers here in atoms and molecules to tissues that they could be centimeters, and in cells we can have micrometers. And I'm sure that you are not familiar with this type of measurements. You might have studied them in high school, or in you remember them in your introductory biology. But if not, we are going to cover it very quickly. The most important that I want that you remember is the micrometer here with the symbol. Yeah, the mu is called a micron and is 1/1,000,000 of a meter, or 10 raised to the power of -6 meter. Okay, and if you remember this, there are different type of measurements that are expressed in that way. So let me remind you some of those measurements. So here there is a scale of all the measurements that we use that maybe you remember from physics, from high school. Here the one that you need to remember are these ones, these three. Nanometers, that are this one, that is 10 raised to the power of -9. Micrometers, that we talked in the previous slide, that is 10 to the power of raised to the power of -6, and this one that is raised to the power of -3, that is millimeter, that I'm sure that you're aware of that because you have it in your rulers in the tiny divisions here between centimeters. Okay, the other ones, we generally don't use it in cell biology. Here is a diagram from your textbook. and here are the measurements that we generally use. So here we use, here you see some nanometers, and here that are, it could be lipids and proteins, and on the other side we have embryos that are, the largest one are in millimeters. Okay, so now let's talk about each one of these measurements and what type of microscope will allow us to see those ones. Just as a reminder, the largest embryos will be one of these tiny divisions of your ruler. Okay, so this is why we need microscopes. In this diagram of your textbook, they talk about two main microscopes. There are many more, but here you can see that you have, for the tiny structures, the electron microscope, and for the other one, you can use a different type of light microscope to observe it. Okay, so we're going to focus on the light microscope in this slide. And in the light, with the light microscope, if you remember in your high school or introductory biology course, you were observing some bacteria, and that are just few micrometers in diameter, and the plant cells that are 10 or 20 times larger. And, and if you remember, those ones, the bacteria is of the size of an organelle of the cell. If you remember then those symbiosis theory, we think that maybe the mitochondria and the chloroplast come from, have a bacterial origin. So this is why you see that these are have comparable sizes. Okay, so this is to study all the cells with different type of light microscopy is enough. However, to study smaller structures, yeah, molecules like lipids and proteins, we will need to use different microscopes. So the light microscope is not enough. We will need to talk about other type of microscopy like electron microscope that are very useful. And we will talk about that later. And remember, this is using nanometers. That is 1/1 billion of a meter. And as we said before, is 10 raised to the power of minus 9 meters. Finally, one that we are not going to use a lot in this course are Armstrongs. Yeah, so are the very tiny ones that, for example, 0.1 nanometers, yeah, is that is an Armstrong, is the size of a hydrogen atom. So this will be very important to measure the hydrogen pump. Yeah, very useful to simulate how these proteins work. And although it's becoming very more frequent to use this type of measurements to study how proteins fold and how they work. For this course, we're not going to use it very often. Okay, and with this, we finished the different measurements that we have in cell biology. So now let's try to answer this question. And this question is based on what we just learned. Which of the following is the smallest biological structure? So here you have 5 structures. You can stop the video or you can continue and think with me about it. Here, you might not remember the picture, yeah? In the test, you might not remember a picture, but you can have a very good guess. So if you just think about, you use all the things that you learned in previous courses and that you learn in this class. So let's start with C, prokaryote cells. So prokaryote cells, we know that is small cell, yeah, smaller than the eukaryotes, but there is not a eukaryote cell. So a prokaryote cell has proteins and have ribosomes that are organelles. So this one is definitely is larger than a protein and a ribosome. The mitochondria has a similar size that a prokaryote. So this will be wrong. If you remember from introductory biology, they have proteins. The mitochondria have proteins in the membrane. So no. If a virus, if you remember, there are different type of virus. There, some of them they can be very big, but let's think about the bacteriophage, the one that attack the bacteria, that are ones that are um, are very popular. Yeah, so that they have here the DNA or RNA here inside. Yeah, but here in this area, okay, let me put this in. Uh, let's see if I can change the color here. You have here a structure that is a capsule where the DNA is inside. That structure generally is lipids and proteins. Yeah, so definitely a virus will be bigger than a protein. Yeah, it's formed by proteins. So so then we only have two options, proteins and ribosomes. And here, or you have to flip the coin, or you can think about this. The ribosome is formed by two structures, and here the translation occur, and here the protein will live. Yeah, the process of translation. So generally, to hold it properly, you will need that the ribosome generally has to be bigger than the protein to be able to do the translation properly. Okay, so I think at least we were able to remove most of them. The between protein and ribosome is start getting a little bit difficult, but here you can see that the protein is around uh, 10 nanometers, while the ribosome will be between 20 to 30. So let me show you that in a second. So here you can see the answer. The answer will be protein, and the protein will be here, as I said before, and it will be the protein will be around 10 nanometers. And here is the answer. Yeah, so here you can see that in your textbook they said that a ribosome, yeah, it could be between 25 to 30 nanometers here, depending if it's eukaryotes or prokaryotes. They can have slightly different sizes, but here you can see that and that they are bigger. That generally the proteins that they could be from 1 nanometer to 10 nanometers, depending on their function and the structure, but generally the proteins would be smaller than the ribosome. Okay, so hopefully that was clear and you're ready for the test. So to summarize this section, we study that biological structures have different sizes, yeah, and different shapes, and we need different microscopes. So there are some that we can see with our naked eyes, yeah. So you can see a person hands with your naked eye, but we also need to observe cellular structure with different type of microscopy, light microscopy and electron microscopy. Now we're going to talk about different type of microscopy in the following slices and I hope that you enjoy it a lot. We are going to use some of this microscope in the lab, and over the semester we are going to um, get into more in detail about the physics and about the different details about this microscope, and in particular how to use it inside of a lab. However, here this is a good introduction of generalities about this different type of microscopy. Okay, we just finished this section of cellular dimensions, and now you know all the different shapes and sizes that cells can have. Now we're going to move into how we study these very tiny cells and what type of microscopies and what type of tools we use to study such a small type of cells. Okay, so with this I finish this video, and in the next one we finish this 1.3 in talking about microscopy.